I work on malaria, which is which remains one of the most important infectious diseases in the world. It's caused by protozoan parasites. So these are, one-celled eukaryotes. They're more complicated than bacteria and viruses of the genus Plasmodium. And these parasites are transmitted to humans by Anopheles mosquitoes. There are 5 species of Plasmodium that cause disease in humans, with the most severe disease, with Plasmodium falciparum being the species that causes 98% of the mortality. There are about 500 million cases of malaria a year and over 800,000 deaths a year due to malaria, and most of these are in children living in sub-Saharan Africa. The GAR Key Project really tells us how far we can get with current interventions in areas where the force of transmission is much higher than it was in the temperate regions. And the GAR Key Project really ran in the early 70s, and it had 2 pillars. There was residual indoor spraying with insecticides, and there was mass drug administration. And they did this for a few years, and they kept incredibly good records, both before and after the project. And it's one of the best epidemiological studies on malaria control that we've had. And they decreased the prevalence of parasitemia, of parasites in people, by 99%. They decreased morbidity and mortality significantly, but they could not interrupt transmission. And when they stopped mass drug administration and insecticide residual spraying, it took about 3 to 5 years for for transmission to come back full force. And I talk about the GAR Key Project, which was a while ago, because the records were so good and so precise. But really, we have had sort of similar situations in many parts of sub-Saharan Africa more recently. So malaria infection is initiated when an infected Anopheles mosquito injects sporozoites. As you will see, they're injected into the skin, and they have to reach the bloodstream to go to the liver. They must go to the liver in order to develop into the next life cycle stage. So these sporozoites invade hepatocytes and then divide and have a huge replication rate. One sporozoite will divide into 5,000 to 10,000 individual merozoites. This stage, these are called pre-erythrocytic stages, because as I will show you, not very many sporozoites are inoculated by a mosquito, and not very many of those that are inoculated get out. And so this is a time when parasite numbers are low in the host, and it's completely asymptomatic. You have no idea that you have been infected. However, once these mature liver stages rupture and release merozoites, they infect the blood, and then in the blood you have these iterative cycles of replication. And this is responsible for all the clinical symptoms of malaria. And now parasite numbers go through the roof. They're on the level of 200 billion parasites or more. I will also mention that a mosquito has to get infected again, right? So there's 2 ends of this. The mosquito infects us, and then we infect the mosquito. And some of these blood stage parasites differentiate to sexual stages, which are taken up by a mosquito. And then these develop in the mosquito to ultimately lead to sporozoites. And so that's the cycle. So once sporozoites are in the skin, they have to move, and they are capable of a motility they are capable of moving, and they move quite fast. And Christine Hopp in my lab made this movie. So this is, again, the ear of a mouse. The mouse has been inoculated with antibodies that label the vessels. So you can see the vessels here in pink. These green things are just autofluorescent hair follicles. And you can see that sporozoites just move around the skin. They look kind of aimless, but and they may be. But when they contact blood vessels, they they sometimes know that they have contacted blood vessels. And actually, Christine has found that their movement, she's tracked their movement and analyzed their movement and found that it changes when they get near blood vessels. So they actually do recognize blood vessels. And here's another movie from Christine. Let's see. Where you can see sporozoites entering the blood vessels. So here's a sporozoite, and it's definitely not coming off the vessel. It probably recognizes that it's in the right place, and it takes a little time, and it does seem to take time for this, and then boom, it goes. So sporozoites get inoculated into the skin. They must be able to move in the skin to find blood vessels and get out. Some sporozoites, a proportion actually, also get picked up and go to the lymphatics, but these never make it to the liver. They never actually generate a productive infection. Although, as Fidel has shown, they can prime the immune response. The immune response. So, in summary of all the sort of data I showed you, I think we can say that the skin phase of malaria infection is a bottleneck for the parasite, that motility is required for sporozoite exit from the dermis, and that parasite proteases regulate sporozoite adhesion. It regulates, in the case of trap, removal of trap from the surface by a rhomboid protease disengages adhesive interactions and enables motility. And in the case of CSP, removal of the N-terminus exposes an adhesive domain and enables the parasite to switch to an invasive mode.